immunity. We were talking about the second line of defense, which is your blood cells. We talked about all the different types of white blood cells and how they function in each type of infection. Okay, so we ended here. We talked about phagocytosis and how phagocytosis occurs. This is a very simplified diagram of phagocytosis. Now, the second protective mechanism is what is called inflammation. Now, we are all familiar with inflammation. If a cut gets infected, you know that place swells up, it's painful. So, inflammation is a process which helps to contain the infection. It doesn't allow the infection to spread. Okay? So, inflammation usually is brought about by the chemicals that are released when a tissue is damaged. So, whenever the tissue is damaged, you will have a lot of chemicals getting released. And these chemicals lead to the signs of inflammation. So, when you start your clinical rotations and all that, or when you start studying pathophysiology, you will learn about this. What are the 5 cardinal signs? Cardinal means most important and will be present in all cases. 5 cardinal signs of inflammation. Five cardinal signs of inflammation are, in fact, the actual words are in Latin, but it is redness. So you will see that that area becomes red. Redness, pain, swelling, heat. So that region of the skin that is inflamed will be hot to touch, slightly warmer than the other part. Heat, and finally difficulty in moving that region, immobility. These are the 5 principal signs of inflammation. And a lot of times when we see patients in the hospital also, if a part is swollen up, we kind of look for all these things. We touch that region of the skin and we touch another region. So, and you have experienced all of this. So, inflammation helps to limit the spread of the infection. Now, how does this inflammation occur? So, whenever there is a breach of the skin, if pathogens enter, because this is a second line of defense, so it has crossed the skin and it has entered inside. When it enters inside, the bacteria are going to multiply, and then tissue damage is going to occur because the pathogen may also cause lysis of the cells. So when the cells break open, they release a lot of chemicals like histamine, bradykinin, and serotonin, prostaglandins. There are lots of chemicals that are released. And these chemicals serve to attract the white blood cells. This is like a signal to the white blood cells that there is an infection going on. So the white blood cells start moving towards that region, and the first step is they sort of stick to the wall of the blood vessel. They stick to the wall. That is called margination. Margination. And once they stick to the blood vessel wall, they escape out. They squeeze out through the tiny little openings between the cells that line the blood vessels. That is the endothelium. That's called the endothelium. So this process by which the white blood cells squeeze themselves out is called diapedisis. Diapedisis. Okay, so they squeeze between the cells and they come out, and then of course they start phagocytosing the bacteria. Okay, and this is what finally leads to the inflammation that you are seeing. Okay, so it sort of leads to that infected material getting collected there, which is what is pus, and that will lead to redness and pain and swelling. So you have to remember that when the white blood cells phagocytose the bacteria or the pathogen, the white blood cell also gets destroyed. Okay, So the pus that you see, which forms during an infection, the pus includes damaged tissue, killed pathogens, and white blood cells. Okay, that's what gives that nature to that pus. So that is another mechanism. Now, any infection is accompanied by fever. So mild, low-grade fever is actually beneficial. Okay, the reason being, high temperature slows down the microbial growth and it also enhances phagocytosis. It helps the white blood cells to do their job. So mild fever is actually good. It is a sign that your immune system, your second line of defense is working. Of course, once the temperature goes above 39, 40 degrees, then that high temperature may be disadvantageous to your own body cells. Okay, enzymes won't work, the cells cannot work. So high temperature can be deleterious, but mild low-grade temperature is actually beneficial. Okay. Now, the next one, we are still in the second line of defense. We talked about white blood cells, we talked about fever, inflammation. Now, there is another set of protective substances which are circulating in your blood. These are called complement proteins. Complement proteins or the complement system. These are proteins, roughly about approximately 20 different proteins. 20 different proteins circulating in the blood. They are found in blood. And obviously, like any protein, liver is the one which synthesizes these proteins. Liver makes these proteins. Okay. So these complement proteins, they've all been named. Okay, they've all been named. So they sort of work in a cascade fashion. That means the first protein will activate the second one. Okay, and then the second one will activate the third one, and so on. So they work like a waterfall effect. So as long as there is no infection, these proteins are not active. They are not going to do anything. But the moment there is an infection, some of these chemicals that are released will start activating the complement system. Okay, and it's a complex process. There are many ways of activation. Your textbook describes it, but at this stage you don't have to know all the steps because each one is different. Now, it's enough, suffice to know that these complement proteins, they activate one another in a cascade-like fashion. Once they activate, what is it that they can do? Okay, what are these complement proteins capable of doing? There are two things that they can do. One, the complement proteins, once they get activated, they can directly cause destruction of the pathogen by forming tiny little pores on the cell wall of the pathogen so that substances will leak out or water can get in and the pathogen can be destroyed. So direct lysis of the pathogen, that's the first thing. Okay, that's how they work. This is possible, direct lysis of pathogen. That's one way. The second mechanism by which complement proteins function is they can coat the pathogen. That is, they can form a coating on the pathogen. They can sort of stick to the surface of the pathogen and make it easier for the white blood cells to recognize which cell has to be destroyed. So they function as a helper. So if you have a pathogen, the complement proteins can come and tag the pathogen like that. So now the white blood cell will know which cell needs to be destroyed. Okay, so that is what this is. So they can tag them. Okay, and this phenomenon or process by which a complement protein sticks to a pathogen we call this, it's a very general term, it's called opsonization. Opsonization. That is forming something like an ID tag that you wear. So if you have to go into a government building, you can't just walk in. You need to have your ID tag. So this is like the complement protein is like an ID tag. So it tags the pathogen so that the white blood cell knows which one needs to be destroyed. Yes. Okay, so that is about complement proteins. And then the last bit, every cell has the ability to produce certain chemicals that are anti-germicidal. Okay, sorry, they are germicidal. That means they can get rid of pathogens. But they are not produced in large amounts. So your cells, once it is infected with a virus, once a virus enters the cell, each cell has the capability of making a chemical called interferon. Interferon, there are many subcategories of interferon, interferon alpha, beta, gamma, you have different types. Now these interferons are basically antiviral substances. Okay? Interferons these are naturally made by your cells when the cell gets infected with a virus. Okay? So the interferons are antiviral. So if a virus particle enters the cell produces interferons and the cell gets destroyed along with the virus. So it is protective because it doesn't allow the virus to spread. Okay, so the cell destroys itself along with the virus. But these are made only in very small quantities. Okay? They are antiviral and they are also anti-cancer agents. They also prevent undue multiplication. But unfortunately, the cells make only very small amounts of this. So if there is a severe viral infection, then it may not be sufficient. But nowadays, interferons, we use interferons for treating viral infections, for treating cancers as well. So these interferons are made using recombinant DNA technology. So we use E. coli and yeast to make it. So it is available as a drug. So you can give it in large quantities. But naturally produced in your body is very little. Iron binding proteins. So last time when we looked at bacteria, we said bacteria release substances called siderophores that will capture the iron which they need. Our body also has many proteins that trap iron, that hold on to iron. So we have transferrin, lactoferrin, ferritin, hemoglobin. You all know about hemoglobin. So these are all iron binding proteins that we make. Okay? And this helps. So once the iron is bound to these proteins, the bacteria do not have access to iron. So it is like a competition. Bacteria produce siderophores to trap iron, and we have a lot of iron binding proteins. And lastly, antimicrobial peptides. So cells also produce small peptides which can have antibacterial properties, antimicrobial properties. For example, it can prevent cell wall from being synthesized. It can make holes in the cell wall to destroy the pathogen. Okay? But these are again produced in small quantities. Now we come to the adaptive or specific immunity. I've already given you the intro to this. So there are two basic subdivisions in adaptive immunity. So let me remind you again about that. Antibody mediated immunity or humoral immunity. So this is adaptive or acquired. So remember, this is acquired after birth. You are not born with it. It is not genetically determined, but it is extremely specific. Okay? And it can actually distinguish between self-antigens and non-self-antigens. And this is further subdivided into two categories. One is humoral immunity or antibody-mediated immunity. Antibody-mediated. The second one is cellular. The second one is cellular or cell-mediated. Cell-mediated immunity. So remember the difference between these two. Humoral or antibody-mediated immunity is towards extracellular antigens. Extracellular, that is, which is outside of the cell. Extracellular antigens and circulating antigens. Anything that is circulating in blood. Whereas this one, cellular or cell-mediated immunity, is towards intracellular antigens, that is, pathogens that are inside the cell. But whenever it comes to a specific infection, both these arms of immunity will work together. Okay? Both of them will work together. It doesn't mean that for one particular type of infection only this will work, and for another one only this will work. The proportion will be different. If there are a lot of extracellular circulating pathogens in the tissues, then this will take the upper hand. Okay? If it is more of a viral infection, then this will take the upper hand. But doesn't mean that that one is not going to do anything. They are both linked. We'll see how they are both linked. Okay? Now this humoral or antibody-mediated immunity is a function of B lymphocytes or B cells. This is a function of T lymphocytes or T cells. Now, where are these lymphocytes produced? These lymphocytes are produced, the stem cell that gives rise to the lymphocytes is in the red bone marrow. Okay, you know all blood cells are made in the red bone marrow. So this is in the red bone marrow, the stem cell. Okay, it's called the lymphoid stem cell. Okay, the stem cell is called lymphoid stem cell or lymphopoietic stem cell. So this cell then gives rise to lymphocytes in the bone marrow. Okay, within the bone marrow, it gives rise to lymphocytes. Lymphocytes are very simple cells like that. Cell membrane and a big round nucleus. That's a lymphocyte. That's how a lymphocyte will look like. Okay. Now, some lymphocytes migrate to the thymus and mature in the thymus. These are called the T cells. So they migrate from the bone marrow to the thymus where they mature. Another population of lymphocyte remains in the bone marrow, and matures there. These are called the B cells. Okay, so they remain there. And once they mature, once they are formed, these B cells and T cells will travel through the blood and lodge themselves in all the organs that we looked at. The tonsils, the lymph nodes, the spleen, everywhere. They'll go and sit there, keeping a watch as to what is coming into the body through the blood or through the tissues, okay, or through your food and air. So they are constantly monitoring. So this population of cells, your bone marrow is constantly making these. They mature in the thymus and then they go and lodge themselves in all the organs of your immune system to keep a watch. Okay? So of these two, the one that matures in the thymus is the one that is responsible for cell-mediated immunity. The one that is in the bone marrow is responsible for antibody-mediated immunity. Okay, so we will look at briefly at these two, what exactly it entails. Before that, what are the key features of acquired immunity? The basic characteristics, something which is very important, and it differs from your innate, inborn immunity. Acquired immunity is very specific. Specific in the sense that it will specifically recognize an antigen, okay, and react with it to destroy it. So the attack pattern in acquired immunity is a very targeted one. It is not a generalized attack pattern. It is a very targeted attack pattern. So specificity is the first characteristic of acquired immunity. Second, memory. Once an attack pattern has been launched against an antigen, that attack pattern will be remembered years later. It will still remember it because adaptive immunity has a memory component. Okay? So which is not the same with inborn immunity. You don't have a memory pattern there. Here you do have a memory. And lastly is what is tolerance. Tolerance is that it is capable of recognizing self-antigens and not destroying your own cells. It only attacks the pathogenic foreign antigens. It doesn't attack self-antigens. And this is a very important characteristic of adaptive immunity. We still are not completely sure as to how this happens, because all of this happens at the fetal stage itself. Even before the baby is born, this happens. Okay, so as these lymphocytes are being formed and as they mature during the, after the baby is born and then it's exposed to the different types of antigens, the cell, any T lymphocytes or B lymphocytes which are capable of recognizing a self-antigen and destroying it, those cells are completely knocked off. They are destroyed. They are not allowed to even multiply. Okay? Only T lymphocytes and B lymphocytes that have the ability to recognize foreign antigens will be allowed to multiply and exist. So the immune system gets rid of any cells which might recognize self-antigen. So it is a fascinating thing how this happens. Immunity, as I told you, immunology is a separate branch by itself. Okay, so how this happens? So there are so many chemicals and things which direct the cells that are going to recognize self-antigens get destroyed. So when the baby is born, the immune system actually already has B cells and T cells that are programmed to identify the innumerable antigens that exist in our environment. How that happens is still not a very clear, we don't know why. But the B and T cells that are there when the baby is born already have the ability to recognize the different antigens which are there in the environment. Even before the child is exposed to the antigen, the cells are already there. Okay? So when you look at the immune system, when you look at these lymphocytes, you will see that there will be populations of B cells. These are B cells. Each one capable of recognizing a different antigen. So I'm just going to, diagrammatically do this, but every B cell and T cell has receptors on the surface. Okay, these are called B cell receptors. So one B cell may have a receptor like this. Another B cell may have a receptor like this. Another one may have something like that. So there are small populations of B cells and T cells, the same thing, each having a different receptor capable of recognizing a particular antigen. So this one will probably recognize an antigen which is maybe like a triangle like that. This one may recognize an antigen which is a circle like that. This one may recognize an antigen which is something like that. So antigens, I told you, are either proteins or lipids or sugars. So each one is capable of recognizing a different type of antigen, and this is done even before the child is exposed to environmental antigens. So the baby is born with some of these cells. So that's the beauty. How does the immune system know what kind of antigens are there? So there are millions of populations of this, okay, small clusters. So the immune system is not going to make too many copies of this, but there will be a small cluster of, let's say, 1,000 cells with this kind of a receptor, another 1,000 cells with this kind of a receptor. So anytime the body gets exposed to a particular type of antigen, that particular B cell will be able to recognize it. Okay, so that is what is meant by tolerance. Okay, so any cell which has a receptor that will destroy our own cells, that gets immediately, it is taken off, not allowed to multiply at all. So these are the three most important characteristics. So specificity, memory, and tolerance. So let's start with a brief description of humoral immunity or antibody mediated immunity. One over there, the one in yellow, is the antigen. That is our pathogen which is coming with the cell wall proteins or whatever. So you have this whole population of B cells in your lymph nodes or in your tonsils, okay, scattered all over. So when this enters the skin and the tissues, when the antigen enters, the population of B cells which has the correct receptor for recognizing, so this is the B cell, these are called B cell receptors. So the correct B cell which is capable of recognizing that antigen will bind to it. The first thing is binding of the B cell to that antigen. The other cells will not, other B cells will not be able to do that because they don't have the right receptor for binding that antigen. So that this is where it is the specificity comes in. So the specific B cell which has the receptor to recognize the antigen will go and bind to it. And now only this population of B cells will start multiplying and making more copies of this. So when you start off, you have a small number of a little bit of each. Okay? And then when this one recognizes the antigen, this particular B cell starts to multiply and make more copies because you need a lot of this to go around looking for the antigen and destroying it. So this is called clonal selection. So a particular clone of B cells is selected by the antigen and then it multiplies. So it is clonal selection and proliferation. Proliferation means increase in numbers, multiplication. Okay? So this particular one will multiply. There's no point in causing multiplication of all the other clones because they are not going to be able to help with getting the destroying the antigen. So this particular cell multiplies and more numbers of this particular B cell comes up, and as they multiply, one group of those cloned B cells will convert themselves into or differentiate into a larger cell called a plasma cell. Okay? And these plasma cells will start secreting antibodies. They will start secreting the proteins which can destroy that antigen. So the specific antibody will be released. So the B cells directly cannot make the antibodies. They have to change themselves into what is called plasma cells. And then these plasma cells start secreting antibodies into the blood. So this is for taking care of the infection that is currently going on. Okay, because these antibodies have to go around in the blood looking for that pathogen with that antigen and destroying it. Another population of that multiplied B cells, they will remain like that and they will go into a memory bank. They will form the memory B cells. Okay, and they will remain. So now this is the first time. The first time the body is exposed to an antigen, the specific B cell, which has the right receptor for recognizing the antigen, will start multiplying. Out of the population of multiplied cells, some of them will convert themselves into plasma cells and start secreting antibodies into the blood. Another population, another small group of those same multiplied cells will become memory B cells. Okay? So these memory B cells are going to help when the individual is exposed to the same antigen a second time. Okay? So this is like a data bank which stores the memory. Okay? So this is humoral immunity. Now let's see what happens with these memory cells. Okay? We'll come to this. Now what is this memory? I keep talking to you and telling you that acquired immunity has a memory. What is this memory? So let's say this is the first exposure to an antigen. Let's just take anything, chicken pox or whatever, okay? Exposure to a bacterial antigen or a pathogen. The first time the child is exposed. Now, what happens? The B cell has to recognize, then it has to start multiplying, then it has to produce antibodies. So for this to occur, it takes at least a week, okay? 5 days to a week for the antibodies to start getting secreted. So first exposure, it may take about 5 to 7 days before you are able to take a sample of blood and see antibodies in it. Okay, you can detect antibodies in the blood only by about a week. And then as the days progress, the amount of antibodies that are being produced will start increasing. And once the infection is completely cleared, the antibody numbers will start decreasing. Okay, once the child is cured, the antibody numbers in the blood will start decreasing. This is the first exposure. Now let's say after many months or even after years, if the child is exposed to the same antigen a second time, this is called the second, this is primary exposure of first one, this is the second one, secondary exposure to the same antigen. Now you will see that within 24 hours, you will be able to detect the antibody in the circulation, and within a day, the amount of antibody that is being produced will be much, much higher than what was done on the first exposure. This is because of the memory cells, because now they know the attack pattern, they know exactly what needs to be done. So the antibody titer, we call it the antibody titer, the amount of antibody will steadily increase within the first 24 hours, okay, and reach high levels. And then as the infection subsides, the antibody levels will come down. And you may still have some circulating antibodies, which will tell you that this person has been exposed to this infection. But let's say this child is exposed to a second new infection. Same process again. So for every antigen or every pathogen that you have been exposed to from your childhood, you can imagine how many memory B cells you must have formed, okay? So every time the individual is exposed to a new infection, this process will happen, okay? is what we saw here. Okay. What do these antibodies do? So the B cell itself is directly not targeting anything. It's not getting, destroying the pathogen. It is these antibodies that are doing it. Now what can these antibodies do? Antibodies can destroy antigens in a number of ways. Okay. One is they can do direct lysis. Antibodies can directly cause lysis of the pathogen. Now, each type of antibody is different, how it functions in a particular infection is different, but this is the general mechanism. So just to look at the overall picture, they can directly cause cytotoxicity. That means they can destroy the cell. Okay. Or antibodies can attach themselves to the pathogen just like the complement proteins. They can attach themselves to a pathogen, making it easier for the white blood cells to phagocytose it. So they can do that as well. So opsonization, they can coat the pathogen just like the complement protein, and they can ensure that it gets destroyed. That is the second mechanism. Third, they can stick to many bacteria at the same time, and they can cause what is called agglutination. They can clump all of them together. Okay? They can clump all the pathogens together, and then that gets destroyed. Or they can, along with the complement, because this is similar to complement protein, along with complement proteins, antibodies can also ensure that the bacterium or the pathogen gets lysed. So they can work in tandem together with the complement proteins. They can also do that. Okay? And sometimes they can neutralize the pathogen. Neutralize means they can stick to the pathogen before it adheres to the tissue. So the pathogen will not be able to attach itself to the tissue and cause damage. So even before it can cause damage, antibodies can stick to the pathogen and they can neutralize them. Okay? So this can be for bacteria, this can be for viruses. So these are the general mechanisms. Okay? So direct lysis, neutralization, agglutination, working with the complement proteins, or opsonization, sticking to them and enhancing phagocytosis. Okay? So this is B cell immunity. Now, when we look at T cell, you will see how these two are connected. B and T cell immunity, both of these are connected. Now, a little bit about antibodies, which you need to know. Because when you go into your clinical years, you will see a lot of tests that we do look for antibodies in the blood, especially infectious diseases. You will look for antibodies. Blood sample will be taken and you will look for the type of antibody that is there. Okay? So when you look at antibodies, this is the general structure of antibodies. An antibody looks like this. It's a protein. Please remember, antibodies are proteins. Okay? Nothing else. So antibodies. Another name for antibodies is immunoglobulins. Immunoglobulins. Or IG. That's the short form, IG. Now these are always proteins. Okay? And they're actually large proteins. Okay? They're large proteins. The structure of an antibody, whenever you look at a textbook or anybody writing about it, they'll always draw an antibody like this. It is a Y-shaped protein which has two heavy chains, a pair of very heavy chain proteins, and two light chains. Okay, the lighter one is the light chain, the heavy one. So the shape of the Y. Now this part of the Y, okay, the arm of the Y, the lower portion of the arm of the Y, and the stem. This is the stem of the Y. This is called the constant region. So this will not change in the, each type of antibody will have a constant part. Now this arms of the Y, the upper portion of the arms of the Y, this is called the variable region. The variable region is what will bind to the antigen. So this will be different. For every antibody, this will be different because it's going to bind to a different antigen. So the variable region is the antigen binding site. Okay. Now there are 5 different types of antibodies. I'm going to tell you something and you have to remember that. IgG. Okay, this is one. IgA, immunoglobulin A. IgM, IgE, IgD. Okay, so.
Is the M the lowercase?
Not lowercase, all uppercase. All of them are uppercase. You never write immunoglobulin with lowercase. All uppercase letters. Okay? IgG, IgA, IgM, IgE, IgD. IgG will have this characteristic shape like this. It's called a monomer. IgE is also like this. IgD is also like this. So each antibody is different. This is just for IgA will look like this. It's a dimer. So it has 2 like that and then joined together. IgA, and then there'll be another thing here. This is the shape of IgA. IgM is a pentamer. That means there are 5.
Do we have to know that part?
No, this is just so that this is how IgM will look. Okay. So they are each different. All of them, if you take IgG, this constant region will be the same for all IgGs. And then the top part, the variable region which is going to bind to the antigen is going to be different, depending on which antigen it is binding to. So each one of them has a different constant region. So there are 5 different constant regions. IgG will have the same constant, this will have the same. Okay? But more than that, because this is more like, if you go into biochemistry and all that, molecular level, you have to know the shape and all. But from a clinical point of view, what is it? IgG is one of the most abundant antibodies. For most infections, IgG is what is formed, okay? Most abundant antibody circulating in blood. So you will find this circulating in blood. Roughly 80% of the antibodies that circulate in the blood are IgG, approximately. Okay? So, no matter what the infection, IgG is the most commonest. The next is IgA. Okay? Roughly about 15 to 20% of the antibodies is IgA. But you don't find IgA a lot in the blood. Okay? IgA is mainly found in secretions, bodily secretions. In bodily secretions, like, think of any secretion, tears, saliva, breast milk, gastric juice, intestinal secretions, vaginal fluid, semen. Any bodily secretion, you will have IgA. Okay? And it is going to be protective. So, all of these antibodies will protect you if anything were to enter. Okay? So that is IgA.
How about sweat?
Sweat also to a certain extent, yes. Sweat as well, yes. Okay, so any secretion you do find these. IgM, okay, now 80% is that, 15 to 20% is this. These are all very little, okay, around 1% or less than 1%, okay, but they are of importance. IgM, this is the very first antibody to be secreted in any infection. In any infection, before the correct antibody is secreted, the first antibody that comes out in the circulation is IgM. Okay? So suppose I'm drawing blood from someone and I do an antibody analysis. Okay? The person seems fairly normal, no symptoms, nothing. But when I test the blood, I find that IgM levels are very high. Then I know that this person is undergoing a recent infection. So they are having an infection. Even though they may not have any symptoms, I know that there is an infection going on because this is the very first antibody to be secreted. So that means the infection is there. It's a recent infection. After a few days, it will change to the specific IgG type. Okay? But the first thing that will be IgM. So when you see an antibody titer with lots of IgM in it, you know that an infection is currently going on. Okay, it's a recent infection. Now IgE, this is the antibody that is responsible for allergic reactions. Allergic reactions. So people who have asthma, chronic asthma, will have IgE antibodies in their blood. Allergic reactions to anything, dust, pollen, medicines, okay? And also worm infections. IgE antibodies will be seen. IgD is actually very, very less amounts, and the function is still unclear. Okay, this one is still unclear. But these are the 5, okay? And I want you to know whatever I have written here. This much you should know for antibodies, okay? And you will, when you go into your clinical rotation and all, you will see reports with these kind of antibody titer, antibody levels. You'll see that. So you should know what the antibodies are. Okay?
With cancer?
Go to cell-mediated immunity. Okay? Now, cell-mediated immunity is a function of T lymphocytes. Okay? Now, T lymphocytes they only recognize intracellular. They are going to fight against intracellular pathogens. So basically viruses or any other bacteria that has gained entry into the cell. So they do not take care of the extracellular circulating pathogens. They are more geared towards intracellular viruses and bacteria. So there are some bacteria that also are intracellular. Okay, they go into the cell. So now lymphocytes, if you look at the lymphocytes, they, just like your B cells produce all these kinds of antibodies, lymphocytes, T lymphocytes, T cells, also produce chemicals. They produce chemicals which we are collectively going to call cytokines. Cytokines are the chemicals that are released by T cells. Okay. Now, in the population of T cells, you have 4 different types of T cells. Okay. And do remember that T cells also have specific receptors like B cells. So if this is a T cell, it will have specific receptors like that. These are called T cell receptors. Okay, T cell receptors. So each particular type of T cell will have a specific receptor. Now there are 4 types of T cells. First is called the cytotoxic T cell. So as the name indicates, cytotoxic, that means it will cause lysis of the cells. This will cause lysis of infected cells. Okay. The way we write this in immunology, they write it as TC, cytotoxic T cell, okay, TC. And in the language of immunology, most immunologists won't refer to it as cytotoxic T cells. They will call this CD8 plus cells. They'll call it this in immunological language. CD8 is the receptor, the type of receptor that is found on the cytotoxic T cell. So they'll always say CD8 cells. Okay. The second type of T cell, second one, is the helper T cell. Helper T cell. Helper T cell, we usually write it as TH. TH, T helper cells. Now, the role of helper T cells, we are going to look at this. They play a central role in immunity. So as the name indicates, they are basically helping immunity, but they play a very central role in immunity. So this is the cell that will connect antibody-mediated immunity and cellular immunity. It connects the 2 arms. This one, helper T cell. Now, helper T cells also, immunologists will not refer to it as helper T cells. They will call this CD4 plus cells. This is how they will address it, CD4 plus cells. The third type of T cell is the memory T cell. Just like memory B cell, every time a T cell recognizes an antigen, you will also have memory T cells being formed. T memory cells. Okay. And then comes the last type of T cell, which is called the suppressor, or some books will also call it regulatory T cell. Suppressor or regulatory T cell. So TS or TR. Okay. Now the function of this suppressor or regulatory T cell is it's like a check and balance on the immune system. So these T cells will ensure that the immune system doesn't get overexcited and start destroying normal cells. So this controls the activity of the cell-mediated immunity. So it ensures that the immune system does not go into overdrive. It doesn't start destroying unnecessary things like normal cells. So it keeps a watch. And this is actually an important role because there is a theory that autoimmune diseases where the immune system starts destroying our own cells, Maybe because these cells are not functioning properly, okay, that they are not keeping a check. So they're destroying even normal tissue, okay. So those are the 4 types of T cells, and all of them secrete different kinds of chemicals, but interleukins and things like that, but we'll collectively call them cytokines. We'll just keep it as cytokines.
I'm sorry, you said for helper T cells, it connects what with what?
The B antibody mediated and the cell mediated immunity.
Okay.
They play a central role, okay. Now this is what, now T cells, because the antigen is inside, okay, try and understand this. So intracellular pathogen, let's say this is an infected cell, okay, and the pathogen enters the cell. Okay. So how will the immune system be able to recognize it? Because you need to have the antigen exposed outside. If it's inside the cell, it is like sitting inside the house with all the doors and windows closed, so nothing can get access into it. So for a T cell to be able to recognize that a cell is infected, this pathogen must be exposed to the outside. Only then the T cell will be able to recognize it. Okay? So whenever a cell gets infected, let's say this is any cell in the body. When a cell gets infected and the pathogen enters, this pathogen will be destroyed by the lysosomes. Okay? And pieces of the antigen, pieces of the bacteria, these are called antigen fragments. These antigen fragments will be exposed on the surface of the cell, on a receptor, cell membrane receptor, because your cell membrane has lots of proteins which function as receptors. So this antigen fragment will be exposed on the surface of the infected cell. So this is an infected cell. When this is exposed like this, now the T cell can recognize it and destroy this cell completely. Okay? A cytotoxic T cell, which has the correct receptor for identifying this type of antigen, will go and attach itself, and then it is going to release a lot of chemicals. Okay? So it will release a lot of chemicals. The T cell is going to release a lot of chemicals, which is going to ultimately lyse the cell. So a cytotoxic T cell can directly cause lysis of the cell. Only thing that is needed is the antigen fragment must be exposed on the surface of the cell, infected cell. Every cell has the capability of doing this. Okay? This receptor, this cell receptor on which the antigen fragment is exposed, this is called the MHC class 1 receptor. It's a protein on the plasma membrane. MHC stands for major histocompatibility. Major histocompatibility protein. Now MHC1 is found on all cells. All cells in your body have this. You might wonder why I'm talking to you about all these details, but I'll tell you the importance of this. Okay? In a brief while, I'll tell you. Knowing about this is important. So every cell in your body has these specific proteins on the plasma membrane on which the antigen fragment can be exposed, so that the T cell, the cytotoxic T cell, can come and bind to it and release a lot of cytokines and destroy the cell. Okay? Now, the same thing can happen in another manner also. Let's say a white blood cell phagocytosis the pathogen. Okay? So So I told you about monocytes, macrophages, which can phagocytose bacteria and other viruses. So these are large cells. These are large cells, a macrophage. And that's the nucleus. Let's say this macrophage has phagocytosed a bacteria. The same process will happen. Okay? The antigen will be destroyed by the lysosomal enzymes. It'll be converted into tiny fragments. And these fragments will again have to be exposed on the surface of the macrophage. Okay? So you have a receptor, and on the surface of this receptor, this antigen will be exposed. Okay? Now these macrophages are also known as antigen presenting cells. Now only difference is this receptor which is found on macrophages is of a different class. That's it, MHC class 2. MHC class 2 receptor. Okay, that's the only difference. So it's a different kind, subtype of the same receptor. Okay. So when a macrophage presents the antigen, something different is going to happen. Okay. When a normal cell exposes the antigen to the outside, a cytotoxic T cell which has the correct receptor to recognize the antigen is going to go and bind, and it's going to release enzymes and destroy the cell. But when this happens, there is a slightly more complex process that occurs. Okay? Now, what is this MHC? Okay? This is very unique class of receptors. No two individuals have the same MHC receptors except identical twins. Okay? None of us will have the same. It'll be different. Okay? Some of it will match, but never the same. Only two identical twins will have the same MHC receptor. MHC class 1 and class 2 will be the same for both of them. Okay? This is the reason why, because we have different MHC receptors, this is the reason why organ transplant is not easy. When you do an organ transplant, can anybody give their organ to anyone? No. What do they do before an organ transplant? They have to do a match. Okay? They do what is called a tissue match. They call the relatives, they call the siblings, and they try to match. And out of all that, many may not match at all. Sometimes a very close relative, they may say, oh, this is about a 60% match. Okay, 60 or 70 is too good. 60 or 70% match, so you will be able to donate the organ to the individual. So what is it that they are matching? They are matching this. This is tissue match, MHC class. You will never get a 100% match. It will never happen. 100% match will never happen unless they are two identical twins. If you get a 60 or a 70% match, that is really good. Okay? So the kidney can be transplanted, the kidney can be donated. The individual can donate it to their sibling. Okay? But then the person who receives the organ transplant has to take immunosuppressive drugs for life. Why? Because once this MHC class protein is different, the individual's immune system will recognize that kidney as a foreign substance, as a bacteria or a virus, and it will try to destroy it. So even after a 60-70% organ match, tissue match, the person who receives the transplant will still have to take immunosuppressive drugs in order for the organ to survive. Do you follow this? So that is the reason. So you can never have two people having the same tissue match. It will never happen. So 60 is kind of really good. You can never have 60, 70. Beyond that, very difficult. And many a times this is what will happen. Okay, most often it is your tissue macrophages or your white blood cells that are ingesting the pathogen, and then they are destroying it, and then they are presenting it on the surface of the class 2 MHC protein. When this happens, the T cell that comes and recognizes the antigen is a helper T cell. Okay, a T helper cell will come and recognize the antigen presented by a macrophage. Okay, and this T helper cell is now, it is not going to be able to, this is an antigen presenting cell, a macrophage already. It is a white part of a white blood cell. So this T helper cell is not going to do anything. On the contrary, once it recognizes the antigen, it is going to release chemicals. It's going to release cytokines, which are going to activate the specific type of cytotoxic T cell which can go around and recognize any cell that is infected with the same pathogen and destroy it. And at the same time, the cytokines that are released by the T helper cell will also activate the B cells, the specific B cells which can produce the antibodies to go around looking for the pathogen in the blood or in the extracellular spaces to destroy it. So that's where you see that both arms of immunity come into effect. So you are trying to take care of cells that are infected with the pathogen. You're also trying to take care of any of the pathogen that is circulating in the blood, or maybe it is in the tissue spaces. So you're ensuring that the attack is on both, not just on infected cells, but also on any circulating pathogen. So the helper T cell is like the link between T cell mediated immunity and antibody mediated immunity. It ensures that both of them get stimulated, okay? So that you're not leaving anything. You're making sure that any infected cell is also destroyed. You're also making sure that extracellular pathogens are destroyed, okay? So that's why we call this the T helper cell as a central role in immunity, okay? So here I would like to tell you that how does HIV, which is the immunodeficiency virus, how is it able to survive, okay, and cause infection, which is like a deadly infection, AIDS, from which patients do not survive once they get AIDS, okay, death usually happens within a year or so. The HIV virus, because it's a virus, it has to enter the cell. It actually hides inside the T helper cell. This is the target cell for HIV, the T helper cell, or the CD4 cells. So the HIV virus, when a person gets infected first, it enters the T helper cell and it remains inside. So it's like having a spy within the army. Okay, you can't recognize unless they are hiding within the army. So your immune system, this is a cell which is supposed to recognize an antigen, but if the virus goes and sits inside that, there's no way the immune system is able to know anything. So the virus remains inside the T helper cell, and every time the T helper cell multiplies, the virus multiplies as well. Okay? And then one fine day, after many years, the virus becomes active, starts multiplying quickly, and causes damage, lysis of the T helper cells. It breaks the T helper cell, goes and infects another T helper cell. So this happens after many years, and once this T helper cell numbers start falling because they are getting lysed, that is when AIDS starts, okay? Because this cell, which is the central one for immunity, this itself is completely destroyed. So there's no way acquired immunity can function once this cell is gone. And that is why the name comes from that, acquired immune deficiency syndrome. So what is deficient? Acquired immunity is deficient. So now you know where the name comes from, because this is acquired immunity. So once the T helper cells are gone, this disease sets in, and once acquired immunity is not functioning, the person is at a high risk for viral infections, bacterial infections, pneumonia, cancers, and it's very difficult to save the individual once it sets in. Okay, so this is what is the gist of this, and this is a slide which shows you the overview of immunity in a very simple manner. When the body is exposed to an antigen the first time, the antigen, if it circulates in the blood, the B cell will recognize it. It will give rise to plasma cells. Plasma cells will produce antibodies which can go around and destroy the circulating pathogen. At the same time, the antigen, the pathogen could be engulfed by macrophages, okay? And they will present the antigen, the helper T cell will recognize it, and the helper T cell will secrete chemicals which will activate the B cell, which will activate the cytotoxic T cells, and then you're going to have the T cells, cytotoxic T cells destroying the infected. Any antigen that has entered the cell will get destroyed. Any antigen that is circulating will be destroyed by the antibodies. And at the same time, you will get memory B cells and memory T cells. So every time you have been exposed to an infection, you can be rest assured that you have in your bank, you have memory B cells and memory T cells, which is protecting you from future infections. So even if you come close to a person who is infected and you breathe in the virus, you're not going to get infected because your memory B cells or memory T cells will immediately destroy them even before the pathogen has a chance to, enter into your tissues or your blood. It will get destroyed. Okay? So that is the importance of memory cells. So this happens all the time, and like I said, depending on what infection it is, if it is a viral infection, cell-mediated immunity will predominate a little bit more. If it is more bacterial or protozoal, then it will be more B-cell immunity. But both of them work. Both of them work together. Okay? So that's it. Now, okay, the last bit is vaccinations. The last bit is vaccinations. Okay? So we can do this. So it's 3:40 now. Let's stop. I'll give you about 20 minutes. Come to the lab. You have to submit the unknown to me today. Don't forget, please submit it so that over the weekend I can grade it, and then I'll put up all your points so far. Okay. Have you done any of the questions that I gave you? Yes. Which ones have you done? Oh, you did all of them? Oh, very good. Which one did you do? I know some of you probably didn't do it. No? Only Esperanza did it?
I did digestive and a little bit of the scan.
Yeah.
Oh, you did digestive?
Yeah, I did all of it.
Oh, you did all of it? Okay, very good. So you like to go over that in the lab quickly?
Yeah.
Should we start with digestive? Digestive. You did skin, you said, right?
Yeah.
Okay.